Study of cell or cell biology began with the discovery of cell by Robert Hooke in 1665. He prepared and studied thin sections of cork of dead plant material under his self-made compound microscope. He observed that cork is composed of minute honeycomb-like compartments, which he termed as cells. According to Hooke, cell is empty space bound by thick walls. Very little information was added to this idea in the following century. The work again started in the beginning of 19th century. Lawrence Oken, a German scientist, in 1805, believed that all living beings originate from or consist of vesicles or cells. Jean-Baptiste de Lamarck in 1805 expressed similar idea and said nobody can have life of its constituent parts are not cellular tissues are not formed by cellular tissues. In 1831, Robert Brown reported the presence of nucleus in the cell. Due to this discovery, Hooke's idea about the cell as an empty space was changed. It was later established that cell is not an empty space. The German zoologist Theodor Schwann in 1839 and a German botanist Schleiden in 1838, working independently, came out with a theory called cell theory. They found that cell consists of three basic parts: nucleus, the fluid called cytoplasm that surrounds the nucleus, and an outer thin covering of membrane called plasma membrane. The cell wall, they said, was an additional structure present only in the plant cell. Keeping in view this definition of cell, the cells could be observed in plants as well as in animals. According to the cell theory, all living organisms are composed of cells or cell products. The cell theory is one of the most fundamental generalizations in biology. It has wide-ranging effects in all fields of biological sciences. Many details of the cells were studied, as a result of which the cell theory was extended. Rudolf Virchow, a German physician, in 1855 hypothesized that new cells were formed only by division of previously existing living cells. It was contrary to the idea of abiogenesis. Living things arise spontaneously from non-living things. One of the prevailing but controversial ideas about the origin of life at that time. Louis Pasteur in 1862 supplied experimental proof of Virchow's hypothesis by demonstrating that microorganisms, means bacteria, could be formed only by existing bacteria. Original cell theory and Virchow's hypothesis gave us the basis of working definition of living things. Living things are chemical organizations composed of cells and capable of reproducing themselves. August Weismann in 1880 said, all presently living cells have a common origin. The salient features of cell theory in its present form are: all organisms are composed of one or more cells. Number 2, all cells arise from pre-existing cells. Number 3, cell is a basic structural as well as functional unit of all organisms. In multicellular organisms, there is a division of labor among cells. Different cells are specialized for different functions. The function of organism as a whole is a result of sum of activities and interactions of different cells and different components of the cell. In animals, for example, muscle cells contract and relax, Nerve cells transmit impulses. Gland cells secrete. Red blood cells carry oxygen. And some stomach cells secrete gastric juice. Similarly, in plants, xylem cells conduct water and mineral salts from soil to the aerial parts of the plant. Phloem cells translocate food. Sclerenchymatous cells support to the plant. Chlorenchymatous cells carry out photosynthesis. Parenchymatous cells store surplus food. Meristematic cells produce new cells for growth and development of plant. As they perform different functions, they show great variations in shape and size. Despite the structural and functional diversity, plant cells as well as animal cells have common plan of organization. The human naked eye can differentiate between 2 points, which are at least 1 millimeter apart. This is known as resolution of eye. The resolution can increase with the aid of lenses. In a typical compound microscope, the resolution is 2 micrometer, which is about 500x that of the naked eye. A compound microscope is a typical laboratory microscope with at least different magnification powers. The typical ocular lenses could be 5x and 10x, but others also exist. Likewise, different types of objective lenses like 20x, 40x, 100x, etc. exist. The magnification power of microscope is determined by multiplying x value of ocular lens and x value of objective lens. Therefore, a microscope with 10x ocular lens and 40x objective lens will have 10 into 40, 400x magnifying power. The resolution will, however, remain the same, which is 500x that of the naked eye. The source of illumination in such microscope is visible light. In electron microscope, the source of illumination is a beam of electrons, and resolution of microscope ranges between 2 to 4 angstroms, which make it 500x greater than that of a compound microscope. Structure of a cell can be studied under light microscope as well as electron microscope. The modern technology enables us to isolate various components of cell, including its organelles, by a process of cell fractionation, and study their structure and function in detail. During cell fractionation, the tissues are homogenized or disrupted with the special instruments. And the various parts of the cells are separated by density gradient centrifugation. The various cellular parts separate out in the different layers depending upon their size weight, and density of the medium. Some cellular components require very high speed for separation from other parts of cell. This is achieved through ultracentrifugation. A cell consists of the following basic components. Number 1. Plasma membrane. Also a cell wall in plant cells. Cytoplasm containing cell organelles. Nucleus with a nuclear or chromatin material. In a traditional system of classification, all organisms are divided into plants and animals. The cells of plants and animals can be distinguished by the presence or absence of cell wall. Cells of animals and plants are complex and have a distinct nucleus. Chromatin material is bounded by membrane and are called eukaryotic. On the other hand, a primitive type of cells such as bacteria lack a definite nucleus and are said to be prokaryotic. In prokaryotes, the nuclear material is directly submerged in the cytoplasm and is not separated from it by membrane. The eukaryotic cells vary greatly in size. They could be as big as an ostrich egg. Most of the cells are microscopic and are not visible to the naked eye. Their size is measured in micrometer. The use of modern technology has made it possible to study the components of cells in details. Plasma membrane or cell membrane is the outermost boundary of cell. However, in most plant cells, it is covered by cell wall. Cell membrane is chemically composed of lipids and proteins. 60 to 80% are proteins, while 20 to 40% are lipids. In addition, there is a small quantity of carbohydrates. Many biologists contributed to establish the structural organization of cell membrane. It was proposed earlier that cell membrane is composed of lipid bilayer, sandwiched between inner and outer layer of proteins. This basic structure is called unit membrane and is present in all cellular organelles. The modern technology has revealed that lipid bilayers are not sandwiched between two protein layers. The protein layers are not continuous and are not confined to the surface of membrane, but are embedded in lipid layers in mosaic manner. This discovery led to the proposal of fluid mosaic model. This model at present is the most accepted one. Cell membrane also contains charged pores through which movement of materials takes place. Transport of materials is one of the vital roles it plays for the cell. It offers a barrier between the cell contents and their environment, allowing only selective substances to pass through it. Thus, it is known as differentially permeable or selectively permeable membrane. The substances which are lipid soluble cross-site more easily than others. Therefore, it regulates the flow of materials and ions to maintain a definite gradient. Many small gas molecules, water, glucose, etc., being neutral can easily cross, while ions being charged particles have some difficulty in crossing. Many substances which are not needed constantly enter the cell by passive transport. Others are taken up against the concentration gradient. They move from the area of low concentration to the area of high concentration. This uphill movement of molecules requires energy and is termed as active transport. The energy used for this movement is provided by ATP. In many animal cells, cell membrane helps to take in material by infolding in the form of vacuoles. This type of intake is called endocytosis, which can be either phagocytosis to engulf solid particles or pinocytosis to take in liquid material. In neurons or nerve cells, the cell membrane transmits nerve impulses from one part of the body to the other to keep coordination. The outermost boundary in most of the plant cells is cell wall. The cell wall of plant cell is different from that of prokaryotes, both in structure and chemical composition. It is secreted by the protoplasm of the cell. Its thickness varies in different cells of the plant. It is composed of three main layers: primary wall, secondary wall, and the middle lamella. The middle lamella is first to be formed in between the primary walls of the neighboring cells. The primary wall is composed of cellulose and some deposition of pectin and hemicelluloses. Cellulose molecules are arranged in a crisscross arrangement. The primary wall is a true wall and develops in newly growing cells. The secondary wall is formed on the inner surface and is comparatively thick and rigid. Chemically, it is composed of inorganic salts, silica, waxes, cutin, lignin, etc. Prokaryotic cell wall lacks cellulose. Its strengthening material is peptidoglycan or murine. Fungal cell wall contains chitin. cell wall is very important. It provides a definite shape to the cell and keeps it rigid. It doesn't act as a barrier to the materials passing through it. The living content of the eukaryotic cell are divided into nucleus and the cytoplasm. The two collectively form protoplasm. Cytoplasm consists of aqueous ground substance containing a variety of organelles and other inclusions such as insoluble waste and storage products. The soluble part of cytoplasm is called cytosol. It forms the ground substance of cytoplasm. Chemically, it is about 90% water. It forms a solution containing all fundamental molecules of life. In the cytosol, small molecules and ions may form true solutions, and some large molecules form colloidal solutions. Colloidal solutions may be sol, non-viscous, or a gel, viscous. Peripheral parts of the cell are often like a gel. The most important function of the cytoplasm is to act as a storehouse of vital chemicals. It's also a site for certain metabolic processes such as glycolysis. In living cells, cytoplasm contains several cell organelles such as endoplasmic reticulum, mitochondria, Golgi complex, nucleus, ribosomes, lysosomes, and centriole. The free-floating cell organelles, such as mitochondria, move about in cytoplasm due to cytoplasmic streaming. This is an active mass movement of cytoplasm. Endoplasmic reticulum. Under an electron microscope, a network of channels is seen, extending throughout the cytoplasm. These channels are often continuous with plasma membrane and also appear to be in contact with nuclear membrane. These membranes vary widely in appearance from cell to cell. The material present in these channels is separated from cytoplasmic materials by the spherical or tubular membranes called cisternae. The two morphological forms of endoplasmic reticulum are a rough form with attached ribosomes and a smooth form without ribosomes. The rough surfaced endoplasmic reticulum is involved in the synthesis of proteins. After synthesis, the proteins are either stored in cytoplasm or exported out of the cell through these channels. The smooth-surfaced endoplasmic reticulum helps in metabolism of a number of different types of molecules, particularly lipids. They also help to detoxify the harmful drugs. In some cells, smooth endoplasmic reticulum is responsible for transmission of impulses. In addition, smooth endoplasmic reticulum also plays important role in transport. Endoplasmic reticulum also provides mechanical support to the cell so that its shape is maintained. Cell contains many tiny granular structures known as ribosomes. Palade in 1955 was the first person to study them. Eukaryotic ribosomes are composed of almost equal amount of RNA and protein. Hence, they are ribonucleoprotein particles. The RNA present in ribosome is called ribosomal RNA. Ribosomes exist in two forms, either freely dispersed in cytoplasm or attached with rough endoplasmic reticulum as tiny granules. Each eukaryotic ribosome consists of two subunits. The larger subunit sediments at 60S, while smaller subunit sediments at 40S. Two subunits on attachment with each other form ATS particle. This attachment is controlled by the presence of magnesium ions. The ribosomes are attached to messenger RNA through small ribosomal subunit. A group of ribosomes attached to messenger RNA is known as polysome. Golgi apparatus was discovered by Golgi in 1898. This apparatus was found virtually in all eukaryotic cells. It consists of sacs or flattened membrane-bound sacs called cisternae. These cisternae together with associated vesicles are called Golgi complex. Cisternae stacks are continuously formed by fusion of vesicles, which are probably derived by budding of smooth endoplasmic reticulum. Their outer convex surface is the forming phase, while the inner concave surface is maturing phase. The cisternae break up into vesicles from the latter. The whole stack consists of a number of cisternae throughout to be moving from outer to inner phase. Transport vesicles are pinched off at the maturation phase of Golgi apparatus. The arrow shows the direction of flow of protein product synthesized on ribosomes. These proteins are converted into glycoproteins in the Golgi apparatus. Golgi complex is concerned with the cell secretions. Secretions are the products formed within the cell on ribosomes and then pass to the outside through endoplasmic reticulum in Golgi apparatus. The secretions are converted into finished products and are packed inside the membrane for export. For example, in mammals, the pancreas secretes granules containing enzymes that help in digestion. The Golgi complex has a role in formation of these granules. The proteins or enzymes which have to be transported out of the cell pass through the Golgi apparatus. The most important function of this complex is to modify the proteins and lipids by adding carbohydrates and converting them into glycoproteins and glycolipids. Lysosomes are cytoplasmic organelles and are different from others due to their morphology. These were isolated as a separate component for the first time by Dituve in 1949. Lyso means splitting and soma means body. Lysosomes are found in most eukaryotic cells. Any foreign object that gains entry into the cell is immediately engulfed by the lysosome and is completely broken into simple digestible pieces. The process is known as phagocytosis, the eating process of a cell. They are most abundant in those animal cells which exhibit phagocytic activity. They are bounded by single membrane and are simple sacs rich in acid phosphatases and several other hydrolytic enzymes. These enzymes are synthesized on rough endoplasmic reticulum and are further processed in the Golgi apparatus. The processed enzymes are budded off as Golgi vesicles and are called primary lysosomes. Lysosomes contain those enzymes which can digest the phagocytosed food particles. Sometimes under abnormal circumstances, starvation, or as a normal physiological process, the parts of the cell are engulfed by the primary lysosome and digested to generate energy. The lysosomes which eat parts of their own cell are known as autophagosomes. The digestive vacuoles in autophagosomes are also known as secondary lysosomes. They are also involved in autophagy, self-eating. During this process, some old, worn-out parts of the cell, such as old mitochondria, are digested. In this way, the materials of cell may be recycled and cell may be renewed. Their enzymes can also result in degeneration of cell, as may occur during some developmental processes. Lysosomes also release enzymes for extracellular digestion. Several congenital diseases have been found to be due to accumulation in the cells of substances such as glycogen or various glycolipids. These are also called storage diseases and are produced by mutations that affect on the lysosomal enzymes involved in catabolism of certain substances. For example, in glycogenosis type 2 disease, the liver and muscles filled with the glycogen within membrane-bound organelles. In this disease, an enzyme that degrades glycogen to glucose is absent. About 20 such diseases are known these days, which are because of absence of particular enzyme. For example, Tay-Sachs disease is because of absence of the enzyme that is involved in catabolism of lipids. Accumulation of lipids in brain cells lead to mental retardation and even death. Dituve and co-workers isolated in 1965 particles from liver cells and other tissues which were enriched with some oxidative enzymes such as peroxidase, catalase, glycolic acid oxidase, and some other enzymes. The name peroxisome was applied because this organelle is specifically involved in the formation and decomposition of hydrogen peroxide in the cell. These are single membrane enclosed cytoplasmic organelles found both in animal and plant cells. These are characterized by containing hydrogen peroxide, producing oxidases and catalases. They are approximately 0.5 micrometer in diameter. They have also been found in protozoa, yeast, and many other types of higher plants. Plants contain an organelle which, in addition to glycolic acid oxidase and catalase, also possess a number of enzymes that are not found in animal cells. These organelles, called glyoxysomes, are most abundant in plant seedlings, which rely on stored fatty acids to provide them with the energy and materials to begin the formation of new plant. One of the primary activities in these germinating seedlings is the conversion of stored fatty acids to carbohydrates. This is achieved through a cycle, glyoxylate cycle, the enzymes of which are located in glyoxysomes. In plants, peroxisomes play an important role in both catabolic and anabolic pathways. In seeds rich in lipids such as castor beans and soybeans, glyoxysomes are the sites of breakdown of fatty acids to succinate. This organelle is present only during a short period in the germination of lipid-rich seed. And is absent in lipid-poor seeds such as pea. Although vacuoles are present both in animal and plant cells, they are particularly large and abundant in plant cells, also occupying a major portion of cell volume and forcing the remaining intracellular structure into thin peripheral layer. These vacuoles are bounded by single membrane and are formed by coalescence of small vacuoles during the plant's growth and development. Vacuoles serve to expand the plant cell without diluting its cytoplasm and also function as a site of storage of water and cell products or metabolic intermediate. The plant vacuole is the major contributor to the turgor that provides support to the individual plant cell and contributes to rigidity of the leaves and younger parts of the plant. Cytosol contains cytoskeletal fabric formed of microtubules, microfilaments, and intermediate filaments. The main proteins that are present in cytoskeleton are tubulin in microtubules, actin, myosin, tropomyosin, and others. Several cell organelles are derived from special assemblies of microtubules, for example cilia, flagella, basal bodies, and centrioles. The movement of cyclosis and amoeboid movements are because of microfilaments. Whereas intermediate filaments are involved in determination of cell shape and integration of cellular compartments. Microtubules are long, unbranched, slender tubulin protein structures. One very important function of microtubule is their role in assembly and disassembly of spindle structure during mitosis. Microfilaments are considerably more slender structures made up of contractile actin protein linked to the inner face of plasma membrane. They are involved in internal cell motion. Intermediate filaments have a diameter in between those microtubules and microfilaments. They play a role in maintenance of cell shape. Animal cells and the cells of some microorganisms and lower plants contain two centrioles located near the exterior surface of the nucleus. In cross-section, each centriole consists of a cylindrical array of 9 microtubules. However, each of the 9 microtubules is further composed of 3 tubules. The two centrioles are usually placed at right angle to each other. Just before cell divides, its centrioles duplicates and one pair migrates to the opposite side of the nucleus. The spindle then forms between them. They are absent in higher plants. Centrioles play an important role in the location furrowing during the cell division. And in the formation of cilia. Mitochondria are very important organelles of eukaryotic cells because they are involved in the manufacture and supply of energy. They are also known as powerhouses of the cells. Under compound microscope, they appear to be vesicles, rods, or filaments. Under an electron microscope, they show complex morphology, although their number, shape, and internal structure vary widely. Mitochondrion is bound by two membranes. The outer membrane is smooth, while the inner membrane forms infoldings into the inner chamber called mitochondrial matrix. These infolds are called cristae. The mitochondrial membranes are similar in structure to other cell membranes. Detailed studies have shown that mitochondria also contain DNA as well as ribosomes. The presence of ribosomes and DNA indicates that some proteins are also synthesized in them. It is self-replicating organelle. the inner surface of cristae, the mitochondrial matrix has a small knob-like structure known as F1 particle. Mitochondrial matrix contains in it a large number of enzymes, coenzymes, and organic and inorganic salts, which help in several vital metabolic processes like Krebs cycle, aerobic respiration, fatty acid metabolism, and many more. As a result of these metabolic processes, the energy extracted from the organic food is transformed into the energy-rich compound ATP or adenosine triphosphate. Membrane-bound, mostly pigment-containing bodies present in the cell are called plastids. Plastids are present in the plant cells only. There are three main types of plastids: chloroplast, chromoplast, and leucoplast. In photosynthetic plants, there are membrane-bound structures containing a green pigment called chloroplast. The green pigment is an organic compound, chlorophyll, which helps the cell to absorb light energy and utilize it to manufacture food. Chlorophyll molecule resemble the heme group of the hemoglobin, a protein used in the transport of oxygen. The main difference between these two molecules is that chlorophyll has magnesium ions while heme has iron ion as the central atom. Chloroplasts vary in the shape and size with a diameter of about 4 to 6 micrometer. Under light microscope, they appear to be heterogeneous structures with the small granules-like structure as a grana embedded in the matrix. Under electron microscope, a chloroplast shows three main components: the envelope, the stroma, and thylakoid. The envelope is formed by the double membrane, while stroma covers most of the volume of chloroplast. Stroma is a fluid which surrounds the thylakoids. It contains proteins, some ribosomes, and a small circular DNA. It is in this part of chloroplast where carbon dioxide is fixed to manufacture sugars. Some proteins are also synthesized in this part. Thylakoids are the flattened vesicles which arrange themselves to form a grana and intergrana. A granum appears to be a pile of thylakoids stacked on each other like coins. On an average, there are 50 or more thylakoids piled to form one granum. On the layers of thylakoids, chlorophyll molecules are arranged, and that is why granum appears to be green. Each granum is interconnected with others by the non-green part called intergranum. Membranes of grana are the sites where sunlight energy is trapped and where ATP is formed. Chloroplasts are self-replicating organelles. Chromoplasts. They impart colors to the plants other than green. They are present in petals of flowers and in the ripened fruit. They help in pollination and dispersal of seeds. Leucoplasts. They are colorless. In shape, they are triangular, tubular, or some other shape. They are found in underground parts of the plants and store food. Presence of cell nucleus was reported in 1831 by Robert Brown. Its early discovery was undoubtedly due to its prominence in many cells, where it stands out slightly darker than the surrounding cytoplasm. It controls the life and activities of cell. In animal cells, it generally occupies the central space, while in the case of plant cells, it's pushed towards periphery due to presence of a large vacuole. Nucleus may be irregular or spherical in shape. Generally, the cells have one nucleus and are called mononucleate. On the other hand, the cells with the two nuclei are binucleate and with more than two are multinucleate. Nucleus is only visible when the cell is in non-dividing stage. It contains chromatin network and soluble sap-like nucleoplasm. In dividing cells, the nucleus disappears, and the chromatin material in it is replaced by the chromosomes. The heredity material is in the form of chromosomes, which control all the activities of cell. DNA, RNA, and proteins, including enzymes, form the chemical composition of the nucleus. Nucleus consists of nuclear membrane, nucleoli, nucleoplasm, and chromosomes. Nucleus is surrounded by nuclear membrane, which separates the nuclear material from the cytoplasm. The nuclear membrane is actually the nuclear envelope composed of two membranes. The outer membrane is at places continuous with the endoplasmic reticulum, while the inner membrane encloses the nuclear content. The outer and inner membranes are continuous at certain points, resulting in the formation of pores, the nucleopores. The nucleopores allow the exchange of materials between the nucleus and cytoplasm. The number of nucleopores is highly variable. The undifferentiated cells, such as eggs, have numerous pores, about 30,000 per nucleus, whereas differentiated cells, such as erythrocytes, have only 3 to 4 pores per nucleus. Each pore has a definite structure which controls the traffic of substances passing through them. Nucleolus. It's a darkly stained body without any membranous boundary to separate it from the rest of the nuclear material. There may be one or more nucleoli in the nucleus. The ribosomal RNA is synthesized and stored in it. It's composed of two regions, the peripheral granular area composed of precursors of ribosomal subunits, and a central fibrillar area consisting of large molecular weight DNA and RNA. It is the nucleolus where ribosomes are assembled and then exported to the cytoplasm via nucleopores. Chromosomes. Nucleus is often deeply stained with the basic dye because of the chromatin material. During cell division, chromatin material is converted into darkly stained thread-like structures known as chromosomes. Under a compound microscope, chromosomes appear to be made of arms and centromeres. Centromere is a place on chromosome where spindle fibers are attached during cell division. Each chromosome consists of two identical chromatids at the beginning of cell division. Chromatid is the exact replica of chromosome, which are held together at centromere. A chromosome is composed of DNA and proteins. All the information necessary to control the activities of cell is located on chromosomes in the form of genes, which are transferred from one generation to the other. The number of chromosomes in all individuals of the same species remain constant generation after generation. In man, each cell contains 46 chromosomes. There are 8 chromosomes in fruit fly, Drosophila melanogaster, 16 chromosomes in onion, 48 in potato, and 14 in garden pea. The number of chromosomes in normal body cells is diploid, 2n, whereas haploid chromosome number or n is present in germ cells. Like human sperms and eggs have 23, while those of Drosophila has 4 chromosomes. Prokaryotic and eukaryotic cell. Biologists have divided cells into two types, prokaryotic and eukaryotic. The differences between these two types of cells are mainly based upon the structure of their nuclei. Eukaryotes have a very well-defined nucleus in which nuclear material, chromosomes or DNA, is enclosed in double nuclear membrane. In prokaryotic cells, however, genetic material or DNA is without any nuclear membrane covering and is directly submerged in the cytoplasm. Organisms possessing prokaryotic cells are called prokaryotes, and those possessing eukaryotic cells are called eukaryotes. Prokaryotes include bacteria and blue-green algae. Eukaryotes include all other unicellular or multicellular organisms, such as animals, plants, fungi, and protista. Prokaryotic cells generally lack many of the membrane-bounded structures found in eukaryotic cells. For example, mitochondria, endoplasmic reticulum, chloroplast, and Golgi apparatus are absent in prokaryotic cells. Since there is no nuclear membrane, a prokaryotic cell has no distinct nucleus and its DNA molecule is directly suspended in the cytoplasm. Prokaryotes have small-sized ribosomes of 70s compared to eukaryotes of 80s. In prokaryotes, mitosis is missing and the cell divides by binary fission. Because of their simpler structure, it was widely accepted for a long time that prokaryotic cells represent a more primitive stage of evolution than eukaryotic cells. Perhaps the most distinctive feature of prokaryotic cell is its cell wall, bound covalently to shorter chains of amino acids forming peptidoglycan or murine. The entire cell wall is often regarded as a single huge molecule or molecular complex called cytoplasm. The cell wall of plants is generally made up of cellulose and is differently structured than that of a bacterium.